The Edward G. Conklin Award was established in 1995 and is awarded annually by the Society to recognize a developmental biologist who has made and is continuing to make extraordinary research contributions to the field, and who is an excellent mentor and teacher, and who has helped train the next generation of outstanding scientists. So many of you will know Conklin's work, maybe from his studies of the marine snail Crepidula. He was the one who discovered that its eggs divide according to a fixed pattern of spiral cleavage. And he realized he could take advantage of this to follow individual cells and their descendants to their final places in the embryo. He performed, therefore, one of the very first ever cell lineage analyses, and he later extended these studies to embryos of other animals, including ascidians. I'm proud that his first faculty position was at Northwestern, where had he stayed there to today, he might have had his funding frozen. But he spent most of his career at Princeton, where if he were still there today, he might have had his funding frozen. But all jokes aside, he was an amazing and consequential scientist, as is the recipient of this year's Conklin Medal in Developmental Biology, Blanche Capel. Blanche, who unfortunately cannot be here with us today, but sent a talk via video, which I'll play for you in a moment, is the James B. Duke Distinguished Professor at Duke University and an international leader in the fields of both developmental biology and reproductive biology. She has won many awards, including an NIH Merit Award, the Frontiers in Reproduction Pioneer Award, the Society of Reproduction Founders Award, and is an elected member of the American Academy of Arts and Sciences. She has consistently served in the leadership of a number of societies, including SDB, where she was the president. And I have to say that I had the pleasure and the honor of being on the board of directors when she was president, and I learned an enormous amount from her. She has helped organize numerous meetings. I left my other thing down over there, so I'm going to wing it from here on. And has just made incredible contributions to our field. Oh no, it's Marcia's fault. She printed it double-sided, so there you go. I really enjoyed reading her nomination package with the letters from the trainees, who just lauded not just her seminal scientific contributions, but also just what an outstanding mentor and role model she was. But, you know, since I was a bit doom and gloom at the outset of this meeting, and Joaquin has followed in my footsteps today, I thought that Blanche would be the right person to take us in a different take-home message. So she was interviewed a few years back by the editors of the Biology of Reproduction. And in that article, she's quoted as saying, "Getting paid to do science is an incredible gift. Enjoy it. Don't let difficulties with funding, failed experiments, or difficult reviewers prevent you from appreciating what is so exciting about this career." And I think that really encapsulates what Blanche brings to the table every single day. And so it's an incredible honor for her to present to her, even in her absence, the Conklin Medal.
So first, I want to say thank you so much for this award from the Society for Developmental Biology. I'm thrilled to receive the Conklin Prize and never imagined that this would happen to me. And I'm particularly excited because I think Conklin's work was really important to the way my work has evolved. Whereas his mentors were committed to descriptive embryology, Conklin incorporated experimental manipulation of embryos into his work. And I always found this very intriguing. I'm so sorry that I was unable to join you there in San Juan due to circumstances beyond my control. But I am really happy to be able to talk to you about what my work and my career path, which is what I've decided to do today. I became a graduate student at the University of Pennsylvania, and there I joined Beatrice Mentz's lab. This was actually not easy to do because I was the first graduate student Beatrice Mentz and only graduate student Beatrice Mentz had. This only happened because of the intervention of Willis Silvers, who some of you may know is a brilliant mouse geneticist who worked on the coat colors of mouse. And he was a friend of Bee's and convinced her to take me. I was older at the time, so maybe her reservations about taking graduate students were overridden a bit by my age. I think I was the only graduate student B ever had, in part because of her formidable reputation, but also in part because she just didn't believe graduate students were worth her time. But in her lab, I worked on the clonal contributions of retrovirally marked hematopoietic stem cells. This was early days in stem cell biology, and we were very excited to be able to label individual cells with retroviruses and inject them into a mouse that had deficits in their hematopoietic department. So we were able to repopulate the compartment without irradiation and show that these hematopoietic stem cells could give rise to all lineages in the mouse. Bea was very instrumental in teaching me about stem cell biology. She was the most remarkable mentor, truly outstanding. And even though she had this mercurial disposition, I had grown up with a father that was a lot like this, so I had learned at my father's knee how to stay out of the way of most of the problems. And the Bea I knew looked more like the Bea you see in this picture, where she was a generous, giving person who told me so many important things about biology. Both Bea and I were very fascinated by germ cell biology. Germ cells, are the ultimate stem cells. And this always fascinated Bea, and it fascinated me. So for my postdoc, I decided to move to a kinder, gentler place, and I went to Robin Lovell Badge's lab in London at Mill Hill. And this was a really exciting place, as it turned out, just by coincidence. My arrival coincided with the discovery of SRY about a year later. SRY is the sex-determining gene on the Y chromosome. This was a very exciting discovery. The field of sex determination had been looking for SRY for many, many years. And Robin injected an XX embryo with a 14 kilobase piece of DNA carrying the SRY coding sequence and produced Randy. Randy was an XX mouse that expressed SRY and developed into a male. And this caused a lot of excitement around the world, and it was the most exciting place to be in the lab at the time. We filled up with newscasters. Everyone thought we were going to start injecting humans and turning them all into males. So it was a very thrilling time to be in the lab, and I wish all of you have an experience like this sometime in your scientific life. There was loads to do. Here is the model that was really drawn by Anne McLaren back in the early 80s, where she drew a model that the bipotential gonad will express a gene that for a long time was called testis-determining region of the Y. And that gene would be able to convert this bipotential gonad to a testis. And the bipotential gonad otherwise would become an ovary, expressing ovary-specific genes and developing oocytes instead of sperm. Now, finally, at last, we had a genetic switch on the Y chromosome. We could identify the gene. We had a starting point for this pathway. And so this was sort of ripe for further discovery. There were a million things to do because we didn't know how SRY was regulated. I had worked on the transcript for SRY and shown that it was a circular transcript, the first circular RNA anybody discovered, which I discovered completely by accident. And we also needed to know what SRY itself regulated. We knew other people were working on the SRY protein. It was known to bind and bend chromatin. So there were so many avenues to work on. It was really a very exciting time. Personally, I was really interested in how the gonad, this bipotential gonad, could develop into one of two organs. As far as I know, it's the only bipotential organ in the early embryo that is truly where all the cells in the organ have the potential to become either cells in the testis or cells in the ovary. And I was really very interested in how this might work. This is a layout of how we think about sex determination in mammals. It is initiated by the fertilization of an X-bearing oocyte with an X or Y-bearing sperm. But it's not until mid-gestation, when the gonad forms, that the embryo begins to develop as a male or a female organ, either as a testis or an ovary. And this was what really fascinated me the most. How would this magical gene, SRY, work in this early bipotential organ to initiate development of a testis? So when I went on the job market, I serendipitously ran into Dave McClay at a developmental biology meeting in the UK, and he alerted me to a possible job at Duke. And it sounded kind of interesting to me. I had grown up in North Carolina, although I hadn't lived there for 15 years. And so it never really occurred to me that I would end up going back there for my career. But somehow I landed in the Department of Cell Biology at Duke in 1993. I knew almost nothing about cell biology. Mike Sheets was the chair of the department, and as many of you may know, he was a really outstanding cell physiologist. He had developed microscopy and tweezers to manipulate the cell membrane and initiate signaling pathways like SARC. Dan Keyhart was also in the department. Dan was a fantastic Drosophila geneticist and cell biologist, and he had built a microscope to look at dorsal closure in the Drosophila embryo. I was very impressed with his work. And I thought, well, all this brilliant cell biology could really help us a lot in understanding how the gonad works. So I was very excited about joining this department. First thing we did was borrow an antibody against laminin from a colleague in the department, Harold Erickson, and my first graduate student, Jeannie Carole, stained the gonad. And we were just blown away by this beautiful image of the gonad here on the surface of the mesonephros and the mesonephric tubules, all labeled with laminin. This was the beginning of our love of confocal microscopy, which has extended throughout my career. Pretty soon, we had an antibody against the first downstream marker after SRY expression, which is SOX9. And this labeled Sertoli cells in the early gonad that was going on to develop as a testis. And within 24 hours, we could show that these cells, these green cells expressing SRY and SOX9, organized around the germ cells, which were labeled by PCAM, and enfolded them into these testis cord structures. So this is sort of a miraculous change in 24 hours in the XY gonad downstream of SRY expression. So how does this work? How could we possibly initiate these changes this rapidly? And I guess this work was really begun by my second graduate student, Kit Tillman, who worked with a technician, Janet Martineau, who came from, luckily, had just arrived in the Triangle from Tom Jessel and Ellen Dodd's lab. That's wrong. Ellen Dodd's lab at Columbia. And we were very excited to have her as a technician for a while in the lab. And she and Kit put together a system for cutting the gonad away from the mesonephros and recombining it using mice of different colors. This experiment was really built on experiments that were done earlier by Anne McLaren, but her markers were not very clear, and so her results were somewhat confusing. But we had benefited from the development of a Rosa GFP mouse, mainly from Phil Zariano's work, and so we were able to use a green mesonephros combined with a white gonad, and to show that actually cells from the mesonephros migrate into the XY gonad, but not into the XX gonad. So this was a really exciting discovery. And soon after this, Jennifer Brennan joined the lab, and she was able to show that the cells that were migrating from the XY mesonephros into the XY gonad were endothelial cells. This was a surprise at the time because we thought they would be the peritubular myoid cells in the testis, but no, it turned out that they were the endothelial cells. And taking advantage of many of the organ culture methods and the ability to introduce signals, to block or activate signals in culture, Humphrey Yao joined the lab, and he began to make Frankenstein gonad sandwiches, excuse me, using bits of XX and XY gonads to look at what would happen if you sandwiched an XX gonad between an XY gonad and the mesonephros. Other people who followed along these lines were Hao Tang, Andrea Ross, Liz Lin, and Corey Bunce, who all did other experiments looking at how signals were affecting the development of the gonad. And I want to thank David Ornitz, who about the same time called me one day to tell me his FGF9 mutants all developed as female. And of course, I said, well, have you genotyped them? And he said, well, no, but he was happy to send some to us and find out exactly we could look at the gonad, find out what was going on. So we were so excited by this. And this work was picked up by Jenna Schmall, who looked at how proliferation was affected in XY gonads that were FGF9 mutants. And Una Kim, who was also looking at how the FGF9 mutants were affected, and showed that, surprisingly, SOX9 came on as usual in an XY mutant. However, it didn't stay on. It went off. In the absence of FGF9, SOX9 couldn't be maintained in the gonad. This was a really life-changing idea. We hadn't really given much thought to the idea that this very strong upstream genetic pathway between SRY and SOX9 could be disrupted by the absence of a signaling pathway. And this led Una Kim and Leo DiNapoli to look further at what happened to the cells in the FGF9 mutants. We thought maybe they died if they expressed SRY and SOX9 but couldn't be maintained. But no, Leo, using markers that had been discovered earlier by others to label the female gonad, went for BMP2 and follistatin, showed that these markers, which were normally absent in the XY gonad in the middle row, turned on in the FGF9 mutant. And it turned out that what was happening in FGF9 XY mutants is that they were switching to the female pathway. So whereas they had initiated development as a male gonad in the absence of FGF9, they turned down all the male markers and turned up the female markers and initiated development as ovaries. This led to Una to establish a model for what she thought was going on in the gonad, and that was that in the early gonad, there was a combination of signals that were involved in even in establishing the bipotential fate of the gonad. But once SRY came on, it upregulated SOX9 and FGF9 and stabilized the male pathway, whereas in the absence of SRY, the female pathway, which was governed by Wnt4, established ovary development. So we thought a little further about what if this was true, what we might expect to happen. And we decided an interesting idea would be to try to stabilize the female pathway in the XY gonad and see what would come of this. And this was done by Danielle Matuk, who overexpressed beta-catenin, a stable form of beta-catenin, in the XY gonad. And we saw something very similar to what we had seen in the FGF9 mutants. Stabilization of beta-catenin led to the downregulation of SOX9 and the conversion of the gonad to an ovary, which led to the development of female mice here, shown on the right, with ovaries and oviducts and uteri. So this was pretty exciting, showing that we could actually intentionally divert the development of the XY gonad to an ovary. And Samantha Jameson took this one step further. We were inspired by Jonathan Hodgkin's brilliant experiments in C. elegans, where he played around with the sex-determining pathways by eliminating different elements of the pathways. Samantha eliminated both Wnt4 and FGF9 in the same mutants, and this resulted in the rescue of the male pathway. This was really a surprise to us when she got this result. If you eliminate FGF9, you lose the male pathway, but if you also eliminate Wnt4, now you get it back. About this time, Doug Coveney joined the lab, and Doug was very interested in vascular imaging in the gonad. He had set up a system for culturing gonads in which the endothelium was labeled, and he decided to try to image this live, to set up a system to do live imaging on vascular development in the XY gonad. And this is the result of his efforts. Here you see the mesonephros on the bottom and the gonad, XY gonad on the top. And you can see that endothelial cells are moving from the mesonephros into the gonad, forming a vessel on the coelomic surface, and then moving downward into the gonad to integrate into the field of cells that was already there. This was a surprising view of what was going on in the gonad. Very exciting work. I was so thrilled. Doug Coveney came from Marilyn Renfree's lab in Australia, and he now works for Leica, I believe. Very wonderful work that he did in the lab. And Jonah Kuhl followed that up by looking at what happened if you actually blocked the vascular development in the gonad by injecting an inhibitor of vasculature, avastin, that was kindly provided by Regeneron, into the heart, into the beating heart of live embryos prior to dissecting the gonad and culturing it in vitro. And what he found was that if you block the vasculature, you block cord formation. Tony DeFalco joined the lab about the same time. He came from Mark Van Doren's lab, and he was very interested in looking at exactly how the vasculature was inducing cord formation. And he and Jonah together showed that vasculature, as it branches downward from the vessel on the surface of the XY gonad, triggers the proliferation of the surrounding mesenchyme, which then invaginates between the cells that are in the gonad, that is, the pre-Sertoli cells and the germ cells, and leads to the partitioning of the gonad into cord-forming units. So the best we know right now, this is how the de novo cords are formed in the XY gonad. Imaging has always been a big part of the lab, and I'm showing you here on the left an embryo cleared and imaged for the formation of the gonad. This was done by Jennifer McKee and Corey Bunce. And Jennifer was also very involved in imaging how the ovary forms in the embryo. Team Ovary has included along the way Lindsay Barsk, Samina Bustamante, Jen, and Delara and Barcy, who has also came along and investigated this new structure that's attached to the ovary called the rete ovarii over here in red. This is a very large structure, surprisingly large structure that we found attached to the ovary using this approach. So I want to thank all of these people for their brilliant work in studying how the ovary forms. Another big influence on my lab was the arrival of Philip Binfie to Duke. He became the chair of biology, and he set up a systems biology group. And this group was very influential on my thinking and on other people in the lab. For one thing, I acquired a graduate student who was a joint graduate student, Anirudh Natarajan, in my lab and in Uwe Euler's lab, who was in bioinformatics and biostatistics. And Anirudh brought some really great ideas to the lab, including this very interesting graph that he produced using the transcriptome data that had been produced by Steve Munger in the lab. So he mapped the expression changes of genes in the gonad on this XY axis, showing their change in the XY gonad on the Y-axis and their change in the XX gonad on the X-axis. And this was the first time I had looked at the changes in genes in quite this way. But what he showed was that there is a very large group of genes that become ovary specific, not because they're elevated in the ovary, but because they're shut down in the testis. And here you can see how the expression of WNT4 is affected by its downregulation in the testis. And this is even more dramatic for the case of MSX1, where the levels stay about the same in the ovary throughout development, but it's very strongly downregulated in the testis. So this was an exciting way to look at the data that I hadn't really thought about before. Meanwhile, Steve Munger, who came to me from genetics, also got very interested in systems biology, but he wanted to use mouse genetics to understand a little more about sex development. And he performed this study along with Paul McGuiney, also in the systems biology group, looking at a first-order conditional independence analysis of gene expression in the gonad. And using this approach, using inbred strains, he was able to define two subnetworks that operate simultaneously in XY cells, a network driving ovary development and a network driving testis development. So at the bipotential stage in XY cells, both of these networks are running at the same time. One of them, the one that drives male development, is shutting down the one that was going to drive female development. Daniel Matuk also got involved in the systems biology group and working with Greg Crawford, who had been involved in the ENCODE project, performed DNase hypersensitivity studies on pre-Sertoli cells. This was our first look at chromatin organization in Sertoli cells, and it took more than 500,000 cells isolated by fax. This took more than a year for Danielle to collect and to perform these studies. When she went on to form her own lab at Northwestern, she switched to a TAC-seq, which we could perform with a small number of cells. And she repeated these studies at the early stage at 10.5 before the gonad has diverted to testis or ovary development. And at that time, she found that the genes associated with both the male and female pathway are all bivalent, bearing both activating and repressive marks in both XY and XX gonads. They all look the same. But then a couple of days later, once sex determination has occurred, you can see that genes associated with the male pathway are stripped of their repressive marks, and the genes associated with the female are stripped of their repressive marks in XX gonads. However, the genes associated with the alternative pathway continue to retain bivalent marks in XY and XX gonads. So don't we need an H3K27 methyl 3 histone demethylase then if we're going to strip off these H3K27 methyl 3 repressive marks? And now I'm going to switch to another part of the lab that has always worked on sex determination in T. scripta. This is a turtle that belongs to the environmental sex determination class. It is a temperature-sensitive species, and genetic differences, if they exist in this species, can be dominated by environmental influences, sometimes by hormones, but most often by temperature. However, the same sort of pattern remains, that you have a bipotential gonad that forms in mid-gestation, and that gonad initiates development as a testis or an ovary. But that depends on whether the eggs are being incubated at 27 degrees, which produces a testis, or 32 degrees, which produces an ovary. Team Turtle has consisted of a lot of people through the years, but I think it was really initiated by Leo DiNapoli when he joined the lab. Leo was very excited about working on turtles. He had a good friend who was a pilot and used to fly him down to Louisiana to pick up the turtle eggs. And this is a picture of Leo DiNapoli and Maria Makedas, who were flying down to get the turtle eggs in this tiny little plane. When Lindsay Barsk joined the lab and she joined Turtle Team, she said there was no way in the world that she was going to climb on this little plane to fly to Louisiana. And so she figured out a way to get the cells shipped to us. And thereafter, they have arrived in big cartons in the lab in late April or early May. This is Boris Tizak, the most recent member of Team Turtle, opening a box of turtles just shipped to us, and with Jordan Botchroff, a longtime technician in the lab. By 2016, and I'm skipping over a lot of paradigm forming work from Lindsay and others, but by 2016, Mike Shazinski had joined the lab and decided to do a transcriptome analysis of turtle development at the female promoting temperature and the male promoting temperature at various stages. And we picked up a lot of the genes whose expression increased in the male or increased in the female, that were genes that were known to be involved in sex determination in other species. But the most important thing we discovered was this gene KDM6B that showed a male-specific pattern of expression from the earliest stages when the gonad forms. This was really interesting because this was a pattern we might expect of a gene that was controlled by temperature and not by upstream elements of the pathway. And KDM6B is an H3K27 methyl 3 specific demethylase, which removes the repressive marks from genes to activate them in development. So this was pretty exciting to us because we had been doing mouse work in parallel, and we thought, wow, this is just what we need. And Carrie Weber joined the lab and working with collaborators in China, produced these mice that carried RNAi to block KDM6B using a viral infection method. We infected turtle eggs. And what we found was that if you block expression of KDM6B, the gonads in turtle develop as ovaries at the male producing temperature. So they look a lot more like the morphology of an ovary shown here than they do like a testis, which again has testis cords, as in the mouse. Carrie went on to show a connection between temperature and its molecular pathway. Carrie showed that in T scripta at 32 degrees, the female promoting temperature, this leads to an entry of calcium into the cells, which is able to phosphorylate STAT3, which is a known repressor of KDM6B. And KDM6B would otherwise activate the male pathway. So phospho-STAT3 blocks KDM6B from its job in activating the male pathway, but only at the female producing temperature when it's silenced. Could this be the demethylase that we've been looking for in the mouse? And in fact, here's Shannon DuPont hoping very much that this is true at the sex determination meeting in Hawaii, praying with some of the sun gods. And sure enough, this turned out to be true, that loss of KDM6B leads to an ovotestis in XY mice. But to our surprise, this was not due to a delay in the expression of downstream genes, as we had anticipated, but instead to a delay in the activation of SRY itself. So this means that SRY in the mouse and DMRT1 and T scripta are activated by the same histone demethylase, KDM6B. And this is a very unusual link between a mammalian system and a temperature-dependent system that we're very excited about following up. I want to mention that Eleanor O'Neill was also involved in this project, and she has been working also on the early pathways in the gonad that lead to its divergence as a male or female organ. I have not treated the germ cell work in the lab with any sort of fairness because I don't think I have time to go into it. But this is a partial list of the people who've been involved in germ cell biology in the lab. I particularly want to mention Victor Ruthid, who really set up our work, a lot of our work on the DND1 mutants, and Talia Hakovich and Kara Stark, who are still working on this project in the lab. I would also like to thank two lab managers who've been particularly influential, Jordan Botchvaroff, who has managed my lab for 25 years, and Megan Harwood, who's recently come on board to keep us all in line. And I also should thank NSF, particularly Steve Klein, and NIH, in particular Susan Taymans, for their help in funding the work in the lab all these years. And I also want to give special thanks to at least three mentors that were very important to me. One of them is Eva Eicher, and another is Paul Bourgoin, both of whom taught me mouse genetics. These are the strongest mouse geneticists I have ever known, and really did so much brilliant work in this area of mouse biology. And I also want to thank Bridget Hogan, who, in addition to being a really important personal mentor for me, recruited many people to the cell biology department when she became chair, who have really influenced my work, including Michelle Bagnat, Stefano D'Italia, Tata, and Ken Poss, all of whom brought many gifts to the cell biology department and taught me many things about development. And maybe I should thank Emil Vici, who was the mentor for Beatrice Mintz. So I didn't know much about Vici until I was preparing an immemorial for Beatrice Mintz, and I went back to look at what he had done in his lifetime. And what Vici was interested in is the bipotential nature of both the gonad and the primordial germ cells, and how genetic and non-genetic factors influence their differentiation in multiple species. Really, what is amazing to me was that the description of his interests would actually work well as an introduction to my lab webpage. This is astonishing to me because I, of course, never knew Emil Vici, and even Bean never talked about him. But somehow, through her, he must have transmitted his interests to me. I find this really fascinating. Thank you again for this wonderful award. Again, I'm so sorry I can't be there with you, but have a wonderful meeting. Thanks.